occurs in two main stages. The first stage of the process, division of the cell nucleus. It is called mitosis. The second stage, the division of the cytoplasm, is called cytokinesis. In many cells, the two stages may overlap, so that cytokinesis begins while mitosis is still taking place. Mitosis. What events occur during each of the four phases of mitosis? Biologists divide the events of mitosis into four phases: prophase, metaphase, anaphase, and telophase. Depending on the type of the cell, mitosis may last everywhere from a few minutes to several days. Figure 8 through Figure 11 shows mitosis in an animal cell. The first phase of mitosis, prophase, is usually the longest and may take up half of the total time required to complete mitosis. During prophase, the genetic material inside the nucleus condenses and the duplicated chromosomes become visible. Outside the nucleus, a spindle starts to form. The duplicated strands of DNA molecule can be attached along their length, the area called a centromere. Each DNA strand in the duplicated chromosome is referred to a chromatid or sister chromatid. When the process of mitosis is complete, the chromatids will have separated and been divided between the new daughter cells. Also during prophase, the cell starts to build a spindle and fan-like system of microtubules that will help separate the duplicated chromosomes. Spindle fibers extend from a region called the centromere, where tiny paired structures called centrioles are located. Plant cells lack centrioles and organize spindles directly from their centrosome regions. The centrioles, which were duplicated during interphase, start to move toward the opposite ends or poles of the cell. As process ends, the chromosomes coil more tightly, the nucleus disappears, and the nuclear envelope breaks down. Metaphase. The second phase of mitosis, metaphase, is generally the shortest. During metaphase, the centromeres of the duplicated chromosomes line up across the center of the cell. Spindle fibers connect the centromere of each chromosome to the two poles of the spindle. Anaphase. The third phase of mitosis, anaphase, begins when sister chromatids suddenly separate and begin to move apart. Once anaphase begins, each sister chromatid is now considered an individual chromosome. During anaphase, the chromosomes separate and move along spindle fibers to opposite ends of the cell. Anaphase comes to an end when its movement stops and the chromosomes are completely separated into two groups. Telophase. Following anaphase is telophase, the fourth and final phase of mitosis. During telophase, the chromosomes, which were distinct and condensed, begin to spread out to a tangle of chromatin. A nuclear envelope reforms around each cluster of chromosomes. The spindle begins to break apart, and the nucleolus becomes visible in each daughter nucleus. Mitosis is complete. However, the process of cell division has one more step to go. Cytokinesis. How do daughter cells split apart after mitosis? As a result of mitosis, two nuclei, each with duplicate set of chromosomes, are formed. All that remains to complete the M phase of the cycle is cytokinesis, the division of the cytoplasm itself. Cytokinesis usually occurs at the same time as telophase. Cytokinesis completes the process of cell division. It splits one cell into two. The process of cytokinesis differs in animal and plant cells. Cytokinesis in animal cells. During cytokinesis in most animal cells, the cell membrane is drawn towards inward until the cytoplasm is pinched to nearly equal parts. Each part contains its own nucleus and cytoplasmic organelles. Cytokinesis in plant cells. Cytokinesis in plant cells proceeds differently. The cell membrane is not flexible enough to draw inward because of the rigid cell wall that surrounds it. Instead, a structure known as the cell plate forms halfway between the divided nuclei. The cell plate gradually develops into cell membranes that separate the two daughter cells. A cell wall then forms between the two membranes, completing the process. Visual summary. Mitosis. Prophase. The chromatin condenses into chromosomes. The centrioles separate and a spindle begins to form. The nuclear envelope breaks down. Metaphase. The chromosomes line up across the center of the cell. Each chromosome is connected to the spindle fibers at its centromere. Anaphase. The sister chromatids separate into individual chromosomes that are moved apart. Telophase. The chromosomes gather at opposite ends of the cell and lose their distinct shapes. Two new nuclear envelopes will form. Cytokinesis. The cytoplasm pinches in half. Each daughter cell has an identical set of duplicate chromosomes. And the first one was interphase, when the cell grows and replicates its DNA and centrioles. 10.3. Regulating the cell cycle. Think about it. How do cells know when to divide? One striking fact about cells in multicellular organisms is how carefully cell growth and cell division are controlled. Not all cells move through the cell cycle at the same rate. In the human body, for example, most muscle cells and nerve cells do not divide at all once they have developed. In contrast, cells in the bone marrow that make blood cells and cells of the skin and digestive tract grow and divide rapidly throughout life. These cells may pass through a complete cycle every few hours. This process provides new cells to replace those that wear out or break down. Controls on cell division. How is the cell cycle regulated? When scientists grow cells in the laboratory, most cells will divide until they come into contact with each other. Once they do, they usually stop dividing and growing. What happens if those neighboring cells are suddenly scraped away in the culture dish? The remaining cells will begin dividing again until they once make contact with other cells. This simple experiment shows that controls on the cell growth and division can be turned on and off. Something similar happens inside the body. Look at figure 10.14. When an injury such as a cut in the skin or break in a bone occurs, cells at the edges of the injury are stimulated to divide rapidly. New cells form, starting the process of healing. When the healing process nears completion, the rate of the cell division slows. Controls on growth are restored, and everything turns out to be normal. The discovery of cyclins. For many years, biologists searched for a signal that might regulate the cell cycle, something that would tell cells when it was time to divide, duplicate their chromosomes, or enter another phase of the cell cycle. In the early 1980s, biologists discovered that a protein in cells that were in mitosis. When they injected the protein into a non-dividing cell, a mitotic spindle would form. They named this protein cyclin because it seemed to regulate the cell cycle. Investigators have since discovered a family of proteins known as the cyclins that regulate the timing of the cell cycle in eukaryotic cells. Regulatory proteins. The discovery of cyclins was just the start. Scientists have since identified dozens of other proteins that also help regulate the cell cycle. The cell cycle is controlled by regulatory proteins both inside and outside the cell. Internal regulators. One group of proteins, internal regulator proteins, respond to events occurring inside the cell. Internal regulatory proteins allow the cell cycle to proceed only when certain events have occurred in the cell itself. For example, several regulatory proteins make sure a cell does not enter mitosis until its chromosomes have been replicated. Another regulatory protein prevents a cell from entering anaphase until the spindle fibers have attached to the chromosomes. External regulators. Proteins that respond to the events outside the cell are called external regulatory proteins. External regulatory proteins direct cells to speed up or slow down the cell cycle. One important group of external regulatory proteins is the group made up of the growth factors. Growth factors stimulate the growth and division of cells. These proteins are especially important during embryonic development and wound healing. Other external regulatory proteins on the surface of neighboring cells often have an opposite effect. They cause cells to slow down or stop their cell cycles. This prevents excessive cell growth and keeps body tissues from disrupting one another. Apoptosis. Just as new cells are produced every day in multicellular organisms, many other cells die. Cells end their life cycle in one of two ways. A cell may die by accident due to damage or injury, or a cell may actually be programmed to die. Apoptosis is a process of programmed cell death. Once apoptosis is triggered, a cell undergoes a series of controlled steps leading itself to destruction. First, the cell and its chromatin shrink, then parts of the cell membranes break off. Neighboring cells then quickly clean up the cell's remains. Apoptosis plays a key role in development by shaping the structure of tissues and organisms in plants and animals. For example, look at the photos of the mouse foot on figure 10.5. Each foot of a mouse is shaped the way it is partly because the cell between the toes die by apoptosis during the tissue development. When apoptosis does not occur as it should, a number of diseases can result. For example, a cell loss seen in AIDS and Parkinson's disease can result if too much apoptosis occurs. Cancer. Uncontrolled cell growth. How do cancer cells differ from other cells? Why is cell growth regulated so carefully? The principal reason may be that the consequences of uncontrolled cell growth in the multicellular organism are very severe. Cancer, a disorder in which the body cells lose the ability to control growth, is one such example. Cancer cells do not respond to the signals that regulate the growth of most cells. As a result, the cell divides uncontrollably. Cancer cells form a mass of cells called a tumor. However, not all tumors are cancerous. Some tumors are benign or non-cancerous. A benign tumor does not spread to surrounding healthy tissue or other parts of the body. Cancerous tumors, such as the one in figure 10.16, are malignant. Malignant tumors invade and destroy surrounding healthy tissue. As the cancer cells spread, they absorb the nutrients needed by other cells, block nerve connections, and prevent the organs they invade from functioning properly. Soon the delicate balances that exist in the body are disrupted, and life-threatening illnesses result. What causes cancer? Cancers are caused by defects in the genes that regulate cell growth and division. There are several sources of such defects, including smoking or chewing tobacco, radiation exposure, other defective genes, and even viral infection. All cancers, however, have one thing in common. The control over the cell cycle has broken down. Some cancer cells will no longer respond to external growth regulators, while others fail to produce the internal regulators that ensure orderly growth. An astonishing number of cancer cells have a defect in gene called p53, which normally halts the cell cycle until all chromosomes have been properly replicated. Damaged or defective p53 genes cause cells to lose the information needed to respond to signals that normally control their growth. Treatments for cancer. When a cancerous tumor is located, it can often be removed by surgery. Skin cancer, the most common form of the disease, can usually be treated this way. Melanomas, the most serious form of skin cancer, can be removed surgically, but only if spotted very early. Other forms of treatment may use the fact that cancer cells grow rapidly and therefore need to copy their DNA more quickly than do most normal cells. This makes them especially vulnerable to damage from radiation. As a result, many tumors can effectively be treated with careful, targeted beams of radiation. Medical researchers have worked for years to develop chemical compounds that would kill cancer cells, or at least slow their growth. The use of such compounds against cancer is known as chemotherapy. Great advances in chemotherapy have taken place in the recent years and have made it possible to cure some forms of cancer. However, because most chemotherapy compounds target rapidly dividing cells, they also interfere with cell division in normal, healthy cells. This produces serious side effects in many patients. It is one of the reasons why scientists are so interested in gaining a better understanding of the role of the cell cycle proteins in cancer. The goal of many researchers is to find highly specific ways in which cancer cells can be targeted for destruction while leaving healthy cells unaffected. Cancer is a serious disease. Understanding and combating cancer remains a major scientific challenge, but scientists at least know where to start. Cancer is a disease of the cell cycle, and conquering cancer will require a much deeper understanding of the processes that control cell division. 10.4 Cell differentiation. Cell differentiation. Think about it. The human body contains an estimated 100 trillion cells. That's a staggering number, but in one respect, it's not quite as large as you might think. Why? Try to estimate how many times a single cell would have to divide through mitosis to produce that many cells. It may surprise you to learn that as few as 47 rounds of cell division can produce that many cells. The result of those 47 cell cycles are truly amazing. The human body contains hundreds of distinctly different cell types, and every one of them develops from a single cell